So I will present you the first results, a collaborative work performed on pectin assessment and to try the relationship between pectin level and cassava cooking ability. And this work has been performed by Julien Ricci, Léa Ollier, and myself. So just to go back on the cell wall structure, Aliénor already showed you these slides. So remind you that cell wall structure is quite complex. There is a primary cell wall mainly composed of cellulose, hemicellulose, and partly of pectins, the blue strand there. And there is a middle lamella, which is mainly composed of pectins. And it is supposed that in this middle lamella, pectins play an important role because they may explain the cohesive network between the cells in the vegetables cells. Okay, but you have to take into account that there is no covalent bonding, at least between cell wall components, between cellulose and hemicellulose, between cellulose and pectins, and between pectins themselves. But there is a lot of entanglements between those polymers that partly explain the cohesive nature of these cell wall components. Okay, but mainly the hypothesis is that pectins play a major role on cell adhesion because of their presence in the middle lamella, and consequently they play a role also on the cooking behavior because the cell wall structure play a major role in the structure of the material after cooking and during cooking. So this is the main hypothesis we want to test. Structure, the pectin of the structure, it is mainly composed of galacturonic acids with two forms of galacturonic acid, either on acidic form, on red figures there, or on methoxylated form, green. And also there is some neutral sugar on the side chain presented here in yellow, okay? But what is important in this case, in this structure, is that the non-methylated, methoxylated galacturonic acid may have ionic bond linkages between themselves due to the presence of cations, and particularly due to the presence of calcium cations. And this gives a egg box structure which can explain linkages between these pectins, and this can also explain the adhesion between the cell and the cell walls. So this hypothesis is mainly that the X-box model, due to non-methoxylated pectins, play a major role on cell adhesion in the middle lamella and consequently on cooking behavior. And we want to test this hypothesis. So we want to assess the structure of pectin and the level of pectin in our RTB foods. So you have to take into account that pectins are composed of neutral sugars, the yellow figures there, and acidic sugar, mainly galacturonic acids. But galacturonic acid is at least 50% of the pectins, but at final, it is less or by 1% at maximum of RTB on dry basis. It's quite low. By comparison, there is a lot of neutral sugar in our RTB foods, free sugars such as sucrose, glucose, and fructose, up to 15 to 20% in sweet potato, for example. But there is also starch, and starch is up to 70 to 80% by itself. So at a total, there is neutral sugar represent at least 80% dry basis, which is 80 times more than galacturonic acid content. So if we want to evaluate pectins, we need a specific reaction. By chance, there is a specific reaction with MHDP, and this specific reaction, there is a specific response of galacturonic acid, which is at least 60 times higher than for neutral sugar. But 60 times is not sufficient because you have neutral sugar, which is 80 times higher than galacturonic acids. So we have to extract pectin before assessment with MHDP. But this is an opportunity, because if we want to extract specifically one or another type of pectin, for example, non-methoxylated pectins, we can use different conditions of extractions, and we will test different conditions of extractions. So we will test different extraction conditions, because we need to extract due to time and also to pH conditions. For example, we can take into account the beta elimination process, which cuts the pectins only on the methoxylated galacturonic acids. And this we will liberate some pectins in the medium. Another reaction that occurs also is the demethoxylation in the same condition at high pH and high temperature too, and this will transform methylated pectin to free acidic pectins. And another reaction that we can use, okay, is use of cation complexing agent. In this case, this cation complexing agent will suppress the egg box structure and will thus free the pectins that are in acidic forms. So we will test different expression conditions: high temperature, low or intermediate temperatures, addition of cations to suppress solubilization of acidic pectin, high pH in any case to have a beta elimination process. And we can use also a cation complexing agent, and we will use EDTA in this case with different molarity. And after extraction, we need a colorimetric reaction. So we use this colorimetric reaction, which is quite simple in principle. This is acid hydrolysis, but at high temperature and at very high sulfuric acid concentration. Then after that, there is cooling down to ambient temperature and MHDP reaction at ambient temperature too. In principle, this reaction is very simple, but it is very dangerous because of the use of sulfuric acid and at high temperature. Also, the time for cooling also is very important, because you have to cool down. Otherwise, the reaction will continue, and you will have a coloration procedure, which is not the one you want. But you don't want to have a too cold product before MHDP reaction, otherwise the MHDP reaction will not occur. So time is very important for all the three reaction procedures in this procedure. So by chance, we have bought this type of continuous flow analyzer to test the pectin reaction. And in this case, the time of reaction is very is fixed by the flow of the liquid in the tubes. And in this case, we can have a very specific and very continuous flow and very continuous reaction time. And we will see that the results are in this case very reproducible. So this reaction, we can use it by manual, but it's preferably performed by using this type of device. So we have tested this extraction and reaction procedure on 6 cassava samples. 2 of them, for example, have a very high water absorption at a very low cooking time. 20 minutes are sufficient for cooking for these 2 ones, for example. And at the opposite, we have bad cooking behavior cassava samples, which needs more than 60 or 60% for cooking. In parallel, we measured the G prime, which is the firmness of the cassava after 10 minutes of boiling, and you can see that the bad cooking samples here have a very high G prime, which means that they are firm and they need high long time for cooking. So there is a great relationship between cooking time and firmness in any case. So we will use these samples and this result to try to correlate these results of G prime with the pectin expression level. So we will test different conditions, addition of calcium, addition of a complexing agent, high pH, different temperature, different duration of extraction, and different mass volume ratio for extraction. If you see the first result for calcium, we do not see very significant result for addition of calcium from 0 to 0.2 molar ratio. This was surprising because it was supposed to block some of the pectin, so we normally should have a decrease of the extraction with addition of calcium. This was not always the case, and even for these samples, there is an increase of solubilization with calcium, which was quite surprising. So we decided not to add calcium to extract the pectins. Second point, we use a cation complexion, EDTA, and you can see on this figure that clearly there is a large increase of the extraction of pectin by addition of EDTA, at least by 5 times. Okay, and there is optimum of concentration of EDTA for the extraction, which is more or less 0.05 molar ratio. After that, we tested the different temperature conditions, and you see that there is an increase of the extraction yield with the temperature, which is normal. But you can see that at this temperature, at 70 degrees C, there is a change in the order of the cultivar, of the varieties. And this is linked to the gelatinization temperature in this case, because this cultivar here has a lower gelatinization temperature. And in this case, at 70 degrees C, all the starch was gelatinized. There is a high swelling of the cells, and there is a release of the cell of the destruction of the cell structure, and so solubilization of pectin for this particular sample. So this is an interaction of the starch with the extraction of pectin in this case. So we decided to use an intermediate temperature of 55 degrees C, which is lower than the temperature of onset of gelatinization for all samples. So final condition, without calcium addition, we can use EDTA, we can use pH 10, which favor the beta emission process. Temperature 55 degrees C. We tested different duration of extraction, but 60 minutes can be a good compromise. And we tested different mass volume ratio, and this mass volume ratio seems also a good compromise. So final results, we obtained for this 6 cultivar, the extraction only with water is very low in this case, and there is no relationship between the extractability of pectin and the quality of the cassava. And you can see on red, good quality cultivar, on red, bad quality cultivar, on green, good quality cultivar, and there is no clear differences between these two types of cultivar for pectin extracted with water. If we add EDTA, a complexing agent, we cannot see any clear relationship between quality, texture, and extractability of pectins. But if we perform the extraction at pH 10, you can see a rich relationship. And in this case, you can see that the higher the pectin extracted in this condition, the better is the quality of the cassava, and the lower the extractability with this pH, and the lower was the cooking quality. The correlation was quite good with these 6 cultivars. And if we add EDTA, you can see there is a great increase of the extractability with EDTA for the bad cooking cultivars, these ones. And in this case, you have also a relationship between extractability of pectins and cooking quality. In this case, you can see that when you have a more pectin extracted with EDTA, you have a bad quality. And this can be seen also when you perform the difference between extraction with EDTA and without EDTA. So cooking quality in this case is very high when methoxylated pectin level is high. It is when these pectin are extracted at pH 10. And cooking quality is high also, whereas non-methoxylated pectin is low. That is when the pectin are extracted with EDTA. So this is what we wanted to demonstrate, and it is quite good on these 6 cultivars. The hypothesis was non-methoxylated pectins play a major role in cooking behavior, and we validated this hypothesis on 6 cassava samples. We also develop a rapid extraction procedure and assessment of pectin families. 2 extractions are sufficient with a pH 10 for methoxylated pectin, pH 10 plus DTA for total pectin, and by difference you can calculate the non-methoxylated pectin. Reaction with MHDP quite easy to perform, but much more rapid and much more reproducible when using a flow analyzer. And the next step is to validate this study on multiple cassava samples and also on yam samples and sweet potato samples. Oh, okay, that's it. Thank you for your attention.
Thank you very much, Christian, for this very insightful and detailed presentation on this promising work on pectin assessment. And as I said for Alienor's presentation, it was 2 webinars ago, it is really appreciable to have taken time to review the difficulties faced during the protocol development and the different alternatives tested to overcome these methodological challenges. So these proofs of concept that you presented is actually one major step of ours in RTB Foods in the understanding of underlying causes of the diversity of cooking behaviors of RTB crops. So thanks a lot. We now open the floor for questions. I don't see any in the chat box for now. But
I would like to start.
Excellent presentation, Christian. Thank you very much for opening perspective for everybody in the project.
Thank you, Dominique. Yes, Gerard has a question, I think.
Yeah. Thank you very much.
I raised my hand first, but...
Okay.
Come on, Dominique. Come on.
Excellent perspective for all the projects. Thank you very much for this proof of concept. And when you say 1 sample every 2 minutes, what does that mean? Because sample arriving dry, freeze dry in the lab, or also taking all the preparation, extraction? And
because it's 1 sample every 2 minutes is a flow analyzer velocity. After extraction, if you extract 10 samples by 10 samples, you can just analyze 1 sample every 2 minutes. It's only 2 minutes between 2 samples.
Yes, we can talk about medium throughput method if we correlate.
Yes, you can analyze 100 samples per day.
Okay, and my question is, you have done this experiment on cassava and compare with potato works. Is it the same on non-methoxylated and methoxylated pectin? Do we have the same behavior with potato?
With potato, Mark worked a long time on this and evaluate the pectin methylesterase activity and relates this activity with the texture. Yes.
Okay.
But it is the same, more or less the same hypothesis. The main hypothesis is that the non-methoxylated galacturonic acid has a great impact on the texture of the pectin and on the cell wall.
Okay, and are you able to analyze sample from other crops? You will receive sample from the different partner, or how you will do with the logistic of analysis?
Yeah, I don't know about the logistical, but we tested the procedure on sweet potato, and it works very. It's clearly we we did not change anything on the procedure, and it works on sweet potato too.
Excellent. Thank you. Gerard.
Yes, Dominique, thank you very much. Thank you very much, Christian, for your very clear presentation. I've learned a lot through your presentation, and I will have 3 short questions, Christian. Please, do you have an idea of the pectin content of cassava and banana? Do you have a comparison of these 2 crops?
I did not perform a bibliographic study on banana.
Okay.
But I think it's higher in bananas than in cassava.
Okay.
I'm quite sure.
Then the equipment that you use in your lab, how much does it cost?
You really want to know that?
And it's just for my information.
Yes,
it's just for my information.
It's 30,000 euro.
Okay, thank you. And finally, my last question is about the quality of the material where you did the extraction. Was it on fresh, or was it on dry matter, or which type of material did you use?
It was a freeze-dried material.
Freeze-dried material. Do you think there can be a difference between fresh and freeze-dried for pectins?
Yes, because there is some enzyme that can hydrolyze pectin and change the structure of enzyme on pectin during drying and during storage. Yes.
Okay, so you advise to freeze dry before analyzing the pectins, right?
Yes, it's better, but you can dry. The rapid drying also is good, but not too rapid, because you don't want to have a gelatinization process during drying.
Okay. Apart from that, Christian, do you have a simple method that you can advise us to use for pectin evaluation?
You can use the colorimetric procedure with MHDP. You can use it by manual. And for example, I know that Bolanley use it routinely, so you can perform it. It is dangerous, but you can perform it. Yes.
Okay. Thank you very much. Thank you very much, Eglantine and Dominique.
You're welcome. Thank you.
Okay, thank you, Gerard. There's a question from Thierry.
Yes, hello everyone. Thank you, Christian, for the very nice presentation. I wanted to ask you about the continuous flow analyzer. So which protocol of extraction are you going to apply or to use with the continuous flow, the pH10 or pH10 and EDTA or something else?
I will use both of them, because with the first one, we have methoxylated pectin, and with the second one, you have total pectin. And by difference, you can calculate non-methoxylated pectin.
Okay. Looking forward for the results. Thank you.
Okay, you're welcome.
There was a question from Suzanne, but I think it has been answered, because it was the same question as Gerard regarding the type of samples, fresh, dried, and so on. There's one question from Didier, but Didier, I think you can ask your question directly to Christian.
Yes, Christian, thank you very much for your nice presentation. My question concerns the extraction step. How long does this step take, please?
The extraction is we choose to extract for 60 minutes, but in fact, 30 minutes will be sufficient if you are very in a hurry, because the results between 30 and 50 minutes of extraction is almost similar.
Okay, thank you very much, Christian.
You're welcome.
Any other comments or question for Christian? I don't see any in the chat box now. Dominique's applause. Okay.
Any perspective from high throughput methods?
Any perspective from high throughput methods?
Yes. collaboration with Web Package 3 on NIRS?
Yes, of course. Yes, of course. And for example, for YAM, we want to, we plan to develop some analysis for YAM from Guadeloupe, and to try to begin, if possible, calibration of pectin level with pectin evaluation by this procedure, by NIRS, and to relate NIRS values with extraction of pectins. For YAM, at least, yes.
Very good perspective for the project. Thank you very much.
Okay.
Another comment from people working on Cassava. I know there are plenty of you here today attending the webinar.
The breeder, Xiaofei, you are there.
Yeah, thank you,
the
Dominic.
procedure is not only for Cassava, it's for all RTB foods in the project.
Yeah, so this is Xiaofei. So at CIRAD, we are harvesting some trials, especially for cooking quality test. So Thierry is managing that. So we collected the water absorption, the cooking time, the near data. So I'm thinking probably that might be a good set of sample. We can send them to you.
Yes, yes. We have these samples, yes.
Yeah, to get the wet chemistry data, and then we can link the nears with your lab data.
Yes, yes.
So
it's a good opportunity. Yes, we had more than 100 of samples from you, but we will not analyze all the samples, but we'll try to analyze a specific set of samples to try the correlation between pectins types and texture and cooking behavior.
Yeah. Okay, thank you very much.
You're welcome.
Thank you, Xiaofei. It's good to highlight possibilities of collaboration. And this is the work done in collaboration at the end. Any other question, remark? Somebody from the team, maybe?
If people can't freeze dry, is it possible to process sun dry or oven dry sample?
Yes, but your sample will change if the drying is too long because of the action of the enzyme present in the product. You know that there is a pectin metylesterase in the matrix, so the structure of pectin will change during storage and during drying.
One last question for Christian before we stop. No?
So this is Sophie. In your slide, you said one sample every 2 minutes. I believe there might be a lot of time spent for prepared samples. Am I right? So roughly how many minutes per sample? Want to know the throughput in your lab. That don't want to spend.
Yes.
Yeah.
The extraction duration is 60 minutes for the moment. After that, you have a centrifugation, and there is no dilution step. So it's quite easy, and I think you can analyze 100 samples every day.
Oh, 100! Wow, that would be nice. Good, thank you.
Thank you, Xiaofei, for this last question. Thanks, Christian, for taking time to present these preliminary results on this new method. So we are really looking forward for more applications on more samples and on other crops, as you mentioned. So lots of opportunities of collaborations between or within RTB Foods framework.
Okay.
So thanks a lot. Dear colleagues, so we have the greeting seasons ahead. So we see each other next year, basically on January 15th. For a webinar on the relationship between sensory descriptors and texture parameters on boiled plantain. So this will be an important webinar acting as an example for all the crops, all the products studied in RTB foods, and a good example on next steps to be implemented in the objective of assessing sensory descriptors with instrumental measurements. And on behalf of the PMU, I would like to wish you a very pleasant and joyful Christmas. And thanks again for joining our webinars. And we see each other next year for a new season of webinars.